Thank you.
Yeah.
Great. So now we open the floor for people to ask the speakers questions. I guess Peng needs a microphone. Yeah. And Ross has another microphone to run around.
We can share mine also.
That's true.
I can just hand it to you as needed.
Yeah.
Well, also, yeah.
We're going to share the microphone?
Sorry.
Do you want us to share the microphone? Okay, that's enough. Thank you. Yeah, we just share. Okay. So any questions for any of the speakers? Oh, great.
Thanks. Peng, I have a question about the missingness indicator method. You mentioned that it is not appropriate for observational settings. Could you give us a sketch of why? And is it just that there are much stronger assumptions needed, or is there a fundamental sense in which it's not appropriate?
Yeah, this is a good question. So in observational study, it depends on what assumption we want to impose. For example, there are two ways. One way is to assume the standard unconfoundedness, like treatment independent of potential outcomes, given the ideal covariates. The ideal covariates are missing. There's another way to impose assumption as treatment independent of potential outcomes, given the observed covariates and the missing pattern. This is in the Rosenbaum-Rubin paper. There are two different ways to impose assumptions. Actually, this does not work under either assumption. I think. Maybe it's better under the second setting, conditioning on the observed covariates and the missing, only the observed part of the covariates and the missingness pattern. It may work, of course, under some model further. Yeah. Yeah, there were simulation studies in epidemiology showing it didn't work. And our own critique on the second assumption is it's not very scientific because it says for people with different missingness pattern, the confounders are different. So the second assumption is a little weird scientifically.
Can you hear me? Okay. So this question is regarding the gene association studies or multiple populations. So as I understand the problem, you want to associate Y with X using some relation that more or less remains invariant across multiple populations, right? So in machine learning, there has been some recent interest on learning invariant predictors, stable predictors. There has been a lot of proposals. So I was just wondering if any of them would be suitable for your task. The second question, related question I would like to ask is, you emphasized a lot on FDR control and being able to have a procedure for false discovery rate control. But usually these machine learning approaches so far do not have any such guarantees like in the knockoff regime, right, for the lasso problem. So I was just wondering whether you've looked at any of these other things and how crucial is this FDR control for your problem?
Thank you. So the FDR, let me start from the bottom. The FDR control is necessary to gain traction in the genetics community. So geneticists have been playing with statistical ideas for a very long time. It's a field that has experienced in the last 20 years an increase in throughput, but it's a very different field from molecular biology in that, when we started having a lot of data in molecular biology 20 years ago, molecular biologists really had not been used to use statistics, and geneticists instead have always been thinking about their problem hand in hand with statisticians. The founding fathers of the disciplines are shared, people like Fisher and so on. So one thing that geneticists have always been very aware of is that when you look through the genome to identifying a location that it's important for a trait, you have a big problem of look everywhere effect. You have a big problem of multiple comparisons. They started out in a setting where they actually could measure very few things, just like blood groups, for example. And so their presumption was, well, if I see an association between blood group and a trait, I have to be very careful thinking about, is it true or not? Because the chances that this trait and blood group sit really close together in the genome is very small because the genome is very big. And now moving on to the current era, where we can assess genomic variation across the entire genome at millions of locations, they are still very concerned about this. If I see an association, I want to be really sure that this is true, that it's not simply because I have many possibilities to look for. So in fact, the standard in the field is controlling family-wise error rate, which is a little bit of an overkill for problems like the one I'm interested in, which are complex traits. So traits that by definition are influenced by very many locations in the genome. So if you are hunting for, say, 100 or thousands of variants that might be important for the trait, it really doesn't make any difference to you if you make one false discovery or not. So the family-wise error rate is really an overkill, and it's totally meaningful to do false discovery rate control. But to abandon any form of multiplicity control, it's really would be perceived in the genetic field as then we cannot trust your results at all. And rightly so. You know, we hear a lot about reproducibility crisis in science. That's not something that has been concerning in genetics. What have been identified as hit in genome-wide association study tend to be highly reproducible, and they are now going to give up this at any stage in time. Now, you are absolutely right that machine learning in general has become very interested in robust and invariant type of predictions, and I think very rightly so. I do think that if we want to have predictive models that we can deploy, we need to have robustness guarantees that, and I think all of these models are interested for this problem. I haven't studied and tried out many, but you know, going back to this FDR control, these knockoff methods are designed to work with any machine learning algorithm. So we actually have tried to play a little bit with Dominic, one of Dominic's method, and wrap around it the knockoff algorithm to see if we can achieve FDR control while emit from the get-go looking for invariant effects. So, but that's a direction that can be explored much further.
Thank you. Any other questions? I'll ask one. So, I mean, the randomization, does it enter in some sense, knockoff is kind of form of negative control and with some randomization kind of in there. And then in Jennifer's work, do you think there's necessity to go into more inference, like uncertainty quantification in the design, like, or you just do kind of the next level?
I think the uncertainty goes into the predictive model that you're leveraging to do the design, and that is something that, so I didn't, so one of my students co-advised with Mike has just put out a paper on, it's like extending conformal prediction to the case where of like active learning, where the test set depends on the training set. And how can you think about uncertainty there? I don't think that works. Yeah, we have to think about how that directly is applicable to some of the kind of on the ground stuff we're doing, but I do think that's very important to understand the uncertainty there.
And false discovery, I think it's kind of relevant there too, right?
That's an interesting question. It's been a long time since I thought about false discovery. I always think of false discovery rate as giving me like practically for the problems I care about, is giving me like a threshold above which maybe my experimenter might want to go look, or some way to think about that threshold. And so if my collaborator wants to, no matter what, measure 100 things, then I just need a rank order. But if he or she wants to know whether it's worth it to do those 100 things, then I guess some FDR might be useful. Or it could also just be I haven't thought about this in a long time.
I think in the narrow sense, that's what people have been doing, like where to cut out. But I think in general, the post discovery is a more general concept. It's like whatever the experiments you do, you want the yield to be high.
Right.
So without having to say how to do that, but I think that's a little more general concept.
Yeah, I guess if you're doing active learning, like there's like how, I don't know how you think about FDR for active learning, because it's not really a notion of like hits, it's a notion of exploration versus exploitation, right? And helping to figure out how to learn the landscape. And then maybe once you feel like you've learned it, then maybe the FDR kicks in.
And also the range could be different. You can be a lot more relaxed and control at 5%. Like
Yes.
explorations, you can change the threshold. It could be interesting.
Yeah.
There's a question here.
Oh, yeah.
Thank you. I have a question for Peng. So Peng mentioned the missing pattern method and demonstrated that using asymptotic analysis that the missing pattern method is better, has better efficiency. And but you also caution against using that in practice. So I wonder, can you provide a finite sample characterization analysis of these different types of methods, and then so the finite sample analysis can provide better guidance in terms of how to use which one in practice?
Yeah, I think that's a more difficult question in theory, like deriving the finite sample bound on those estimators. And even if we derive those things, usually it depends on a lot of unknown constants. So I'm not sure whether the theory can give us guidance, but I think it's the gap between theory and practice. Really, we prove asymptotics and then looking at the finite data, then we'll just say, okay, do we trust the theory or not? Yeah, I think that's a good question, but it's very difficult to answer using theory.
So I'm going to ask a very similar question here, which is that I guess you'd given us an asymptotic ordering in terms of the efficiency and the asymptotic. So, we're working in a finite population model where asymptotic doesn't mean draw more samples. Asymptotic means I have a new population. I have a sequence of populations and I put conditions on how the sequence is. And, I guess with that ordering, I wanted to clarify and kind of ask a question. I guess the thing I wanted to clarify is I was looking at it, I think, well, to make that ordering true, I guess we have to have an asymptotic sequence where the most complicated estimator, where it's like the missingness of like all pairs and that kind of stuff, and higher order missingness, we have to see enough of the higher order missingness in our asymptotic to make that good. Whereas, if I had a weaker asymptotic, sequence where I said, oh, you'll never observe a lot of pairs or a lot of higher order, but you still observe missingness for the single order, then the estimator that you do say, I think will perform better, because the other one we can't prove anything about. So all that to say is I was looking at this ordering, and I guess my thought was there's no like one true asymptotic or ordering. Like under different asymptotic sequences, the more complicated thing might be worse. Under more conservative ones, or under more stringent, stronger conditions, the more complicated one performs the best. And I think that seemed to capture a lot of like the, which one do we recommend? If we're doing asymptotic analyses, comes down to which asymptotic sequence do I believe in more? And I guess I was hoping, one, to see if I'm on the right page with this, or if I've missed a crucial step, but also how you think about, this fact that comes up particularly in finite population, where it's as I've run into this fact too, it's a very difficult thing to interpret the asymptotics. How do you think about that when we get to choose the conditions on our asymptotics?
Yeah, I think that's a very good question. So here we focus on the design-based theory. So we focus on the finite population effect, but at the same time, we also talk about the asymptotics, saying the sample size grows to infinity. Like people laughed at us, saying, okay, you are using two contradictory words for the same problem. But that's in some sense, it's a challenge for frequentist statistics, right? We have a finite data set. We say, oh, we embed this finite data set into a sequence. Hypothetically, then sequence grow, then we have some properties for that sequence. But whether then those properties tells us something about the finite data we have to make a decision, maybe based on other things, like which sequence is good for us, like which sequence match reality better. In some sense, is beyond the data. I think we need some other guidance. Yeah, we're talking about this issue. We have another reading course, Foundations of Statistics. Like we always face this challenge when do statistics theory, like from finite to infinite. Like deeply, you can say this is related to the problem of induction, like based on n data points. You want to say something beyond this n data points. But the frequentist solution is hypothetical sequence.
Hi, so I have a question for the third speaker. So you were talking about looking, your problem is, roughly speaking, you want to search over a huge discrete space. And you said that in practice, people have some good ideas of what are the good starting points for this directed evolution. May I know, like, is this like some domain knowledge? And how accurate is this starting points? What happens if the starting point is bogus or?
Yeah, there's I mean, it is domain knowledge of different levels. So like one level is just like an actual protein that you already know exists, like for some reason, I guess. So that's quite common. And then other forms of domain knowledge come in terms of, for example, how to induce variation in the population. So I didn't talk about it, but instead of just randomly making changes from a parent to get a population, something you do instead is you actually make like blocks, contiguous blocks, and then you change around the blocks from the different variants, like kind of interchangeably. And there's a question of like, where can I put a breakpoint in the protein that's going to induce useful variation, where useful means doesn't like collapse, so it still functions. And moreover, the function I care about is, has some variability. And so there people bring in domain knowledge from the like structure, for example, as deduced by a crystal structure or AlphaFold. So there is quite a lot of domain knowledge. The other kind of domain knowledge that comes in is just through very long decades of research. People may know that I can change these 4 positions on this protein, and that's just useful for the same kinds of reasons. So some of the domain knowledge is just the actual protein you start with. And sometimes it's a set of parents where you then play like Lego blocks a bit. Sometimes it's a single one, but it is all typically from like many years of work that have revealed domain knowledge. So there's a different version of protein engineering I didn't talk about, but you may have heard of David Baker's lab at the University of Washington, which does biophysics-based modeling, of course, now also with machine learning, but it took a really long time before they agreed to go there. And they're kind of arguing for more what is called de novo design, which I hesitate to use because I don't think it was ever super well defined. And I think it's really not well defined now. But the idea there is to say, I don't really know a starting point. I'm just interested in, can I make proteins that look nothing like existing proteins, but still stably fold? And not even necessarily for a particular purpose. And so in that case, there's still reliance on, let's say, core Lego blocks that exist in all proteins that we call like the secondary structure. You may have seen pictures with these loops, these like alpha helices or these beta sheets. So there's some sort of quite core components and so they're still using domain knowledge, but ultimately it's more biophysics driven as well.
Thank you.
Have a follow-up question on that. So you mentioned the piece X giving theta. What form is actually? Yeah, so I mean, we never did any great due diligence on that. We were using variational autoencoders. And it's not something we ended up exploring very much for the particular AAV gene therapy collaboration. In that case, we used the distribution permitted by the control knobs, which was this very simple
marginal distribution over the amino acids at each position. Because that's all they could control.
So you didn't really go for some non-parametric forms, like from autoencoder, just in the algorithm, you get the form for the p theta. Because you're optimizing over px theta, right?
Yeah.
Over theta.
Yeah.
Did you ever write down an analytical form for that?
Well, I mean, do you call the variational autoencoder analytical?
Well, I'll call algorithmic.
I mean, we played either with variational autoencoders or with the particular distribution that we were allowed to give back to the lab. I know that at some point, I knew some people at Google that were trying to explore more systematically what might be a good model for that. There's so many interacting parts here, but I mean, something I didn't mention is that when you're trying to learn this predictive model that goes from sequence to a scalar property of interest, I mentioned it as though it's primarily from labeled paired data, but actually there's a huge dependence in many cases, not in our example, but on using actually evolutionarily related data that are not strictly speaking coming with a label. So, and then you build a density model from a set of proteins and you interpret the evaluation of the density as the fitness. And then there's a large body of literature there asking what are good suitable model classes to do that and why are they good, and this kind of thing. But that's a slightly different problem because that's saying I want to model a set of proteins that I think are related evolutionarily and capture their density well, whereas we're using it as like a mechanism as part of the design process. And you might ask like, how are those similar and how are those different? I think is actually an interesting question.
Thank you.
I have a question for the genome association study. Although the knockoff method seems to control the FDR, it seems to make much fewer discoveries. So my first question is this because these discoveries are far apart, like in the genome space, whereas the GWAS discoveries are more concentrated among each other? Or is this because the knockoff method is based on conditional independence, whereas the GWAS is based on just an independent test? And the second question is like, if so, like how do we make the trade-offs among the total number of discoveries, the FDR rate, and the total number of populations to consider?
So the multi-environment methods in the examples I showed you makes much fewer discoveries because it insists these discoveries to be consistent across multiple environments. And what happens there is that you have one environment with many observations, so it's easy to make a discovery there. It's very difficult to reject the nodes in the other environment. So you have a drop of power. The moment you insist it has to be in at least two environments, it's just very difficult for you to do that. So it has, it's not the conditional hypothesis that it's a problem. In fact, the comparison methods I showed you are all testing conditional hypotheses. So what's happening in general, when you insist the discovery to be true in multiple environments, you're going to have a higher time making those. And in the particular data set that I showed you, it's really not well powered to make discoveries across multiple environments. I also want to make another point about FDR control that was connected to what was raised before. So Jennifer says, oh, it doesn't really matter. It's a question of ranking my results, and then my collaborator might test as many as they want. And that's certainly true in some setting. It's not true in gene mapping. People do not know how to test if an association is important for a phenotype. I mean, they know in theory, but say to prove that some gene influences your risk of developing schizophrenia, it's very hard to do experimentally. You don't modify embryos and see if they develop schizophrenia or not. So you have to go to an in vitro system. And how do you measure schizophrenia? You know, you can do mouse models, but all of these things take years. So there is that thing to keep in mind, that not all of the type of experiments that we do can be handed off to somebody else to verify the discovery in an effective fashion quickly. I think it's also a question of being able to know, to describe to the world what is it that we know. You know, we do science, we make these discoveries, and yes, there is a rank order among them, but it's important for us to be able to communicate to the public. We believe that this is really true or not, even if we don't have at the moment that we make it, we have the possibility to retest it in 100 different setups. Did I answer your question?
Yes, yes.
I just want to say, I completely agree. And I used to work in statistical genetics and my comment was restricted solely to, to be clear, I completely agree.
I have a follow-up question. Yeah, so one other way to do it is I understand that one cannot verify experimentally whether an association is true or not. So I get the point. So not quickly, yeah. But then putting on my machine learning hat, I would say, well, if I have access to data from 6 populations, I'll withhold the 6th one, right? I'll try to do some invariant prediction of whatever algorithm I have without any FDR procedure on the 5 of them, and then try to see if it holds in some sense on the 6th, right? And I would, if it just works for any one holdout, I would just leave one holdout, and I would just say this is a good and this is a good model, right? Without making any statement about how much false discovery I have. So that's one question I had in mind. The second thing is, if you go into knockoff filters, right, they do assume that you have the ability to generate the conditional distribution of xj given x minus j. I understand that in genetics, maybe the Gaussian model is well known to work very well. If that's the case, probably that's a good thing to, that's a great thing. But in general, if I want to do this knockoff FDR control over arbitrary distributions and non-parametric associations, the main difficulty is to verify this conditional sampling assumption. And you are just offloading the problem somewhere else. So your FDR guarantee is not exactly an FDR guarantee because it's conditional on having this conditional sampler. I know there have been works that actually deal with this problem and all that, but just wanted to say the FDR also has one subtext. So yeah.
Yeah, I mean, there is never, I don't know if this working. There is never any free lunch, thank you. But so for sure you can say, I build a model and then I test it in another population. And in some sense, that's what people do, even what I did in the last table, I have, okay, this is my results and this is how many of these have external validation, right? I don't think that, however, if you're building the model and testing how it works in another population in terms of does it have predictive power, it's not terribly useful to have these test errors to measure how well you selected your variables. I think the evidence that we have accumulated so far, it's really that it's quite difficult to estimate test error to start with, and it's not terribly easy to backtrack from the test error to the validity of the variable selections. Now, to construct knockoff, you need to know, at least the knockoffs I mentioned here, you need to know the distribution of the predictor variables. And that it's a big assumption. At some point I insisted you don't have to make any assumption on how the trait depends on the genetic variants. But what we're making assumption is that we know the distribution of the genetic variants. So it depends, you're right, that depends in the context where you are at. In genetics, I think to make the assumption that we know the distribution of the genetic variants is really not a crazy assumption. We have a lot of data sets that have been collected on genotypes in many populations. We can use them to fit models for the genetic variant. And incidentally, we don't use Gaussian approximations. Typically, we have models that are hidden Markov models that are accepted in genetics for other reasons. But there you have a variety of tools. In some other data sets, you might not have really the abundance of observations on X's to fit proper distributions. Now, there are, as you mentioned, there are some works that look at this problem in more general setups with adversarial networks. And so you can do these deep knockoffs if you have enough data, even if you have no prior knowledge on what the distribution is, you can try to fit it. But that is definitely one reality you have to confront yourself to. Do you have enough data on the axis or knowledge to input there to know what their distribution is? It's also true that if you're looking at your data with models that are fairly simple, like the lasso, in fact, all what you need to match are the variance covariance structures. So even if you want to say I let out, use every possible machine learning tools, then you really have to have knockoff that completely match in distribution your original variable and cross exchangeability. But if you're using simple tools like lasso, really the second order moments is all what matters. So it makes it much easier.
Well, I think this is a great discussion, and let's carry it over to lunch. And let's thank three speakers for 3 wonderful talks. We'll come back at 2 for another session, 3 speakers and a half. Yeah.